Dear students, today I'm going to talk on the topic polyploidy and its significance. These are the contents of today's lecture. The chromosomes of each species has a characteristic morphology and a characteristic number. But sometimes, due to certain accidents or irregularities during cell division, or during crossing over, or during fertilization, some alterations in the morphology as well as the number of the chromosomes that takes place. Such variations are called as chromosomal aberrations. They are also referred to as chromosomal mutations. Now, these chromosomal aberrations are classified into two types, structural and numerical. So, let us look into the various terms. The small x refers to the basic chromosome number, also called as genomic chromosome number. Small n refers to the gametic chromosome number. 2n refers to zygotic chromosome number or somatic chromosome number. Genome means the basic set of chromosomes. Now, in the somatic cells of a diploid organism, two copies of the same genome are present. That is, 2n is equal to 2x, whereas the gametes always contain a single genome. That is, n is equal to x. So, any deviation from the diploid state, that is 2n is equal to 2x state, is referred to as numerical chromosomal aberrations. It is also called as heteroploidy. Now they are of the following types: aneuploidy and euploidy. So let us look into the numerical changes of the chromosomes. So this is a chart regarding the numerical changes of chromosome. So it is categorized into two types: aneuploidy and euploidy. So aneuploidy is that phenomena wherein the variation in the chromosome number occurs by less than one set. This is further categorized into hypoploidy and hyperploidy. Hypoploidy is further categorized into monosomy and nullisomy. Hyperploidy is further categorized into trisomy and tetrasomy. Now let us look into euploidy. So euploidy is a phenomena wherein the change in the chromosome number takes place by one or more sets. So this is categorized into haploidy, diploidy and polyploidy. Polyploidy is further categorized into autopolyploidy and allopolyploidy. So in this talk, I'll be dealing with euploidy, polyploidy and autopolyploidy in particular. Now what is meant by euploidy? EU in Greek, it means even. Ploid means unit. So the term euploidy actually means having one or more complete sets of the chromosomes or the genomes. Now these may be identical to each other or they may be distinct from each other. Now euploid variations, they are designated with reference to the diploid, that is 2x state and not the 2n. Now please look into this table. The first column depicts the euploid. Then the second column shows the number of genomes which are different from 2. And the third column represents the symbol. So in euploidy, the first type is haploid. So it has gametic chromosome complement and it is represented by symbol small n. Monoploid, here the number of genomes, it is having one genome and is represented by the symbol small x. The second type is polyploids. In this, again, it is divided into autopolyploids where the genomes are identical with each other, and allopolyploids where it is having two or more distinct genomes. Now some examples of autopolyploids, that is autotriploid, if it has 3 copies of the same genome, then it is represented by 3x. Autotetraploid, if it has got 4 copies of the same genome, represented by 4x. Autohexaploid, 6 copies of the same genome, represented by 6x. Now in allopolyploid, for example, allotetraploid, it has got 2 distinct genomes and each genome has got 2 copies. That is 2 into the first copy plus 2 into the second copy. Allohexaploid, it has got 3 distinct genomes. So 2 copies of the first genome plus 2 copies of the second genome plus 2 copies of the third genome. Now let us look into what is meant by polyploidy. When an organism has got more than 2 genomes, then that phenomenon is called as polyploidy. It is of the following types. First one is AutoPolyploidy, when all the genomes present are identical. So, for example, capital A. The second one is allopolyploidy. Here there are 2 or more distinct genomes. For example, A, B, C, D. Third one is auto-allopolyploidy. Here, presence of different types of genomes in which one genome always remains as an autopolyploid. So you can see there are 4 copies of A genome. That is, it is autopolyploid by the presence of 4 copies of A genome. And because there are 2 copies of B genome also, it becomes a auto-allopolyploid. Fourth type is segmental allopolyploidy. Sometimes what happens is the genomes that are present are partially homologous to each other. So, for example, A1 genome is partially homologous with A genome. That is why A1, similarly B1, B2, C1, C2. Now let us look into autopolyploidy in particular. So autopolyploids, they are polyploids that receive all their chromosomes from the same species, as we have already discussed in table 1. So if an organism has got 3 copies of the same genome, it is called as autotriploid and referred to as 3x. If it has 4 copies, autotetraploid, 5 copies, autopentaploid, and so on. And all the chromosome sets, they are homologs, and naturally occurring polyploids are very rare. But just look into this pic. So here you can see there are 3 chromosomes and there is 1 copy of chromosome number 1, 1 copy of chromosome 2, 1 copy of chromosome 3. It has been also represented by different colors. There is only one set, so it is called as haploid. It has got 2 sets over here, so this is diploid. Now here there are 3 sets, so this is an example of the autopolyploid. Here there are 4 sets, so it is a autotetraploid. Here it is having 6 sets, that is, it is autohexaploid. Now let us look into how the origin and production of autopolyploids takes place. So it happens by 2 methods: spontaneously, or it may be even induced. So let us look into this one by one. Spontaneously. Spontaneous means when the phenomenon of autopolyploidy occurs without the influence of any external force. For example, sometimes chromosome doubling takes place spontaneously in the somatic tissue. Sometimes even this may occur due to abnormal meiosis, and because of that unreduced gametes are produced. And union of such unreduced gametes, it gives rise to tetraploid zygotes. Now sometimes what happens is unreduced gametes, they are produced because they are under the influence of the action of the gene which causes a synapsis or desynapsis. Now, examples of spontaneous autopolyploidy are in some ornamentals like tulip. It also occurs in hyacinth, and it has also been found to occur in the forage grasses, for example, the ryegrass. Now let us look into the other method of spontaneous production. Sometimes there is a production of adventitious buds. Decapitation occurs in some plants. What is meant by decapitation? So when the apical bud is removed, that is called as decapitation. Now when the apical bud is removed, what happens is callus development takes place at the cut end of the stem. And such a callus, it has got the cells. Some of the cells may be polyploid, and some of the shoot buds which are regenerated from this calyx, it may be polyploid. And example is this is of common occurrence in family Solanaceae, where nearly about 6 to 36% of adventitious buds have been reported to be tetraploid. And if we want to increase the frequency of this tetraploid adventitious buds, then you can apply 1% IAA at the cut end of the stem. Now, the other spontaneous occurrence is the endosperm. So, you are aware that in angiosperm, the endosperm is diploid. So, in most of the plants, endosperm is diploid, and this is formed by the fusion of the male gamete N with the diploid polar nucleus, that is 2N, to form the triploid 3X endosperm nucleus. Now, there are some triploid animals also. For example, Drosophila, salamanders, lizards, and isopods. Now let us look into the other type of um, autopolyploidy. Okay, it can even be induced by means of using the physical agents. So heat treatment or cold treatment or chill treatment, then centrifugation, then X-ray or gamma irradiation. This may also produce polyploids. For example, in case of datura, when the datura plant exposed to was given the cold treatment, it was found that some of the branches were tetraploid. Similarly, when the maize plants or the ears of maize, they are exposed to a temperature of 38 to 45 degrees centigrade, especially at the time of the first division of zygote, nearly 2 to 5% of the progeny produced was found to be tetraploid. Now, heat treatment has been successfully used in barley, wheat, and rye, and also some other crop species. Now, the other method of induced autopolyploidy is by in vitro culture. So sometimes certain plants, when they are regenerated from the callus that has been produced by tissue culture technique, they show autopolyploidy. Example, Nicotiana and Datura. Then by using chemicals. Some chemicals, for example, colchicine, nitrous oxide, sulfanilamide, mercury chloride, dinitroenils, hydroxyquinoline, chloral hydrate, they induce polyploidy. Now these are called as inducers. Now these inducers, they disturb the mitotic or the meiotic spindle and thus cause the non-disjunction. What do you mean by non-disjunction? It means non-separation or non-segregation of the already duplicated chromosomes. Now let us talk about this colchicine treatment. So chromosome doubling or induction of autopolyploidy can be brought about by colchicine treatment. So you can see a pic over here of a plant. You can see this branch. This is the normal diploid branch. When the colchicine is applied to the terminal bud, and what happens is the branches which are formed, they will show the chromosome doubling. That is, it will show the phenomenon of autopolyploidy. You can see over here the leaves are larger in size and also the fruits are larger in size. Now, if you take the cutting of this branch, okay, and then propagate it, then you can get diploid plants. Now, this is a very common method that is followed in Kashmir, where apples are grown, apples and plums. So colchicine treatment is the most effective and the most widely used method for chromosome doubling, and this has been used for a large number of crop species, especially both of dicot as well as the monocot group. Now, what are the different methods of application of colchicine? So, it may be by seed treatment or by seedlings treatment, or even the colchicine can be applied to the shoot buds in case of woody plants, or it can be even applied to the shoot apical meristems, especially in case of grasses as well as cereals. Now please look into this pic over here. So you can see that this is a diploid cell. When it undergoes, that is during the metaphase, you can see that the chromosomes are lying over uh, on the equatorial plane, and when the cell moves on to anaphase first, what happens is there is no spindle formation at all. So all these chromosomes, they come and lie in the same cell. And what has happened is there is no cytokinesis at all. There is no division of the cytoplasm and formation of the cell wall. So all the chromosomes lie in the common protoplasm, and that is why the chromosome number gets doubled. So you see that now this cell has become 4n tetraploid. So the parent cell was diploid, and now the daughter cell has become tetraploid. You can just look into this pic also. So this is a diploid cell with 4 chromosomes. During the mitotic prophase, you can see each chromosome has split to form two sister chromatids. Now, when you subject such cells to colchicine treatment, what happens is uh, that the when the cell moves on to the anaphase, okay, there is no spindle fiber formation. And as a result, what happens is all the chromosomes, they come and lie within the same cell, and there's a doubling of the chromosome number. Here you can see there are 4 chromosomes, but here you can see that there are 8 chromosomes. Now let us look into autopolyploids in animals. They are quite rare. Autoploidy is quite rare in bisexual animals, but it is found in some sexually reproducing animals like earthworms, insects, shrimps, fishes, and amphibians. In such insects, reproduction is found to be usually occurring by parthenogenesis. There is one South American frog, Odontophrynus americanus, with 4x is equal to 44. It does reproduce sexually, but the irregularity is that there is high frequency of quadrivalence, and therefore meiosis is irregular. Now let us look into the genetic effects of autopolyploidy. What are the morphological and the cytological features? Now these autopolyploids, they show larger cell size. The cell size is large. Stomatal features, the guard cells of the stomata, the stoma, they are larger in size. So you can see in this pic, this is a 2N. Okay, this is a leaf surface of the diploid in the diploid condition. The leaf surface of a tetraploid showing tetraploid condition, and the leaf surface of the octoploid. So you can just compare the size of the stomata. You can see the guard cells are quite small. The stoma is smaller. Here the guard cells and the stoma is quite bigger, and in octoploid you can see that the guard cells are quite big, and the stoma, the opening is also quite big. You can also compare the size of the epidermal cells. You compare the size over here, here, and here. So in diploid they're small, in 4N is medium, and in octoploid you can see the epidermal cell size has also increased. Now, but here what happens is the number of stomata per unit area is lower. What about the pollen grains? It has been found that the pollen grains are also larger in size. Now about the flowering period. It's found that the plants that show autopolyploidy, they are slow in growth and they flower late. And what about the size of the leaves and the flowers? So you can see over here in the pic, this is a diploid, whereas this is the autopolyploid, the flower of a diploid and the flower of the autopolyploid. So these are smaller in size. Where are these? These are larger in size. So these autopolyploids, they show less less leaves but larger leaves. The number of leaves are less, but they are larger in size and they are thicker. Similarly, larger flowers and fruits are produced, but they are lesser in number. Now about fertility. Autopolyploids, they show reduced fertility. That is mainly due to the irregularities during meiosis. Then what about the vigor and the growth? In many cases, it has been found that autopolyploids, they this phenomena of autopolyploidy leads to an increase in the general vigor as well as vegetative growth. But in some cases, in some few cases, it has been found that the polyploids are similar to diploids or sometimes they are even weaker. Now, what is the difference in optimum ploidy? It is found that for different species, they have got different levels of optimum ploidy. For example, in Beta vulgaris, that is beetroot, the optimum ploidy is 3x. Whereas for the Timothy grass, Phloem pratense, it is between 8 to 10x. Now let us discuss about cytology in case of autopolyploidy. Autopolyploids, they are characterized by the formation of multivalents in meiosis first. And also, pairing here occurs between more than two homologous chromosomes. Now let us discuss one by one. What happens in autotriploids? So, you can just look into this pic. Here, there are three chromosomes shown. They're all similar to each other. That is, it's a autotriploid. And each chromosome also is showing two sister chromatid. Now, what are the pairing possibilities? Okay, there are three different pairing possibilities. So you can see all these three chromosomes, they may align with each other, and that is called as a trivalent. Or out of these three chromosomes, only two of them may align with each other. So then a bivalent is formed. And when a bivalent is formed, a single chromosome, it represents the univalent. So these are the pairing possibilities in case of autotriploidy. So we found that in autotriploids, either one trivalent may be formed or one bivalent and one univalent may be formed. Now again, in case the bivalent and the univalent is formed, that is, if 2n plus n segregation takes place, okay, then what are the different types? So out of these three chromosomes, hmm, so what may happen, that the first chromosome and the second chromosome, it may move towards one pole, and the third chromosome, it may move towards the opposite pole. Here, or the other possibility is that the first chromosome and the third chromosome may move to one pole and the second chromosome may move to other pole. Or the other possibility is that the second chromosome and the third chromosome, that is cis and this chromosome, may move towards one pole, whereas the first chromosome may move towards the other pole. So you can see there is a large variety of the possibilities. So this probability of obtaining a diploid gamete or a haploid gamete is given by the formula 1 by 2, okay, raised to um, x minus 1. Now, during anaphase first, what happens is disjunction of the trivalents and distribution of the univalents is irregular. What do we mean by this? Now, during anaphase first, what may happen is that out of these three chromosomes, okay, the distribution, these two chromosomes may move to one pole and this may move to the other pole. So the disjunction of the trivalents and also the distribution of the univalents is irregular, and this results in the production of aneuploid gametes, also called as unbalanced gametes. And such unbalanced gametes, they are unviable and they are sterile. For example, in banana and watermelons. Now, let us discuss about autotetraploids. Now, look into this pic over here. There are four chromosomes. That is, it is the autotetraploids, four copies of the same chromosome. Now, what are the pairing possibilities? So, the pairing possibility is that two bivalents may be formed. Okay, one bivalent, these two chromosomes may align with each other to form one bivalent. These two chromosomes may align to form another bivalent. There's another possibility that all the four chromosomes they may align with each other to form one quadrivalent. Or the other possibility is that out of the four chromosomes, three may align with one another to form a trivalent, and one may be separated, that is, it may form a univalent. So, in autotetraploids, you can see that one quadrivalent may be formed, or one trivalent and one univalent may be formed, or two bivalents may be formed. So, what happens during anaphase 1 then? Disjunction of the trivalents and also distribution of the univalents is irregular and resulting in the production of aneuploid gametes. So, what is happening over here? We have discussed at the level of the chromosomes. So, these chromosome assortment, okay, assortment, that is movement towards the poles, it takes place at random. It does not take place systematically, but the chromosome assortment takes place at random. That is, unbalanced gametes are formed. And here, in case of autotetraploids, fertility is variable. Means some are fertile and some are sterile. But fertility has been improved by selection in many species of maize and bajra and rice and rye. Now let us look into segregation, how segregation takes place in autopolyploids. Now when we discuss in case of a diploid, okay, in case of a diploid, if a gene exists in two allelic forms, say capital A and small a, then the number of possible genotypes in a diploid is capital A, or capital A, then capital A, and small a. But the number of genotypes in an autotetraploid, that is 4x level, you can just look into this pic. These are all the possibilities. Okay, so the totally five type of genotypes are produced. So let us look into the five type of genotypes. So you can see capital A, capital A, capital A, capital A. That is, there are four. Okay, there are four alleles over here. There are four dominant alleles, and when four dominant alleles are present, it is called as quadruplex. Now, there is three capital A's and one small a. It is called as triplex. That is, it is having three dominant alleles. If it has got two capital A into small a, that is, it is called as duplex. It has got two dominant alleles. Now there is only one capital A and three small a. It is called as simplex. That is, it has got only one dominant allele. And if there are no capital A at all four are small a. That is, there is no dominant allele. All are recessive. So it is called as nulliplex. Now let us look into what are the type of gametes formed. So, in quadruplex, what's the possibility of the gamete? All the gametes produced will be capital A, capital A. That is, only one type of gametes are produced. Now, let us look into triplex. In triplex, there are two types of gametes produced. One which is homozygous with capital A, and other one is heterozygous with capital A and small a. Now, what happens with in case of duplex? You can see that there are three different types of gametes with homozygous AA, homozygous small a, and heterozygous, that is one capital A and one small a. Now, in case of simplex with only one dominant allele, you can find that there are two types of gametes formed: capital A small a and small a small a. So what happens over here? In case of nulliplex, there's only one type of gamete produced, small a, small a. Now, herein I have taken one typical example. Okay, so during gametogenesis, in duplex autotetraploid, what happens? You can just look into this. Okay, so this is a parallel generation with um, like two dominant alleles and two recessive alleles crossed with another P1, um, that is the male line of the same genotype. So what are the gametes formed over here? You can see that there are three type of gametes are formed: homozygous capital A, homozygous small a, and heterozygous capital A and small a. Now this occurs in the ratio of 1 is to 4 is to 1. Now this is the Punnett checkerboard, and you can just look over here that what is the genotypic ratio now? Where all the four dominant alleles, that is quadruplex, there is only one. Triplex, you can see over here, these are the ratios given. So this is 4 and this is 4. So triplex is 8. Then duplex, you can see the duplex, a duplex over here, they are 18. And in case of simplex, you can see over here 4 plus 4, that is 8. Then in case of nulliplex, it is only 1. So genotypic ratio is 1 is to 8 is to 18 is to 8 is to 1. What is the phenotypic ratio? It is 35 dominant is to 1 recessive. Now we talked about chromosome segregation. Now there are different types of chromosome segregation. The first one is random chromosome segregation. So up till now, in the previous slides which we discussed about the separation or the segregation of the chromosome, uh, that is at the chromosome level, that is random chromosome segregation. Now not only random chromosome segregation takes place, but also random chromatid segregation also takes place, leading to a many increase in the number of the irregularities. Now here is a pic. I just put it up over here so that you can have a look of the karyotype of the real chromosomes of maize. So these are all stained by Fuxen, and this is by Gish. So you can see over here that there are 10 chromosomes in this line over here. That is, the diploid chromosome number of maize is 2 and is equal to 20. So you can see 10 and 10, 20. Again, you have 10, again 10. So this is an example of a autotetraploid. The maize over here is showing autotetraploidy, and these are real chromosomes. Now, what is the mechanism of autopolyploidy? So it takes place by non-disjunction, and this occurs both during mitosis as well as during meiosis. The second method of autopolyploidy is by genome duplication. Now just look into this pics now. So this is a diploid cell which is having two chromosomes. Okay, it's having um, this is one homologous pair of chromosome, and this is another homologous pair of chromosome. So totally there are four chromosomes. Now during the S phase of mitosis, what happens is that the replication of the DNA takes place, and when the cell enters metaphase, you can see that each of these chromosome um, forms two sister chromatids. Now, separation of sister chromatids does take place, and when the cell moves to anaphase first, okay, you can see that the chromosomes have become V-shaped. But what happens is that sometimes these spindle fibers are not formed. So, though the separation of the sister chromatids has taken place, these sister chromatids cannot move to the opposite poles because the spindle apparatus has not formed. So, non-disjunction has taken place or non-segregation has taken place. So, in the same protoplasm itself, in the same cell, all these chromosomes they come and lie in the same cell. So, what happens now is this daughter cell has got 8 chromosomes, and this becomes a autotetraploid. You can just compare this daughter cell with the mother cell. Here it has 4 chromosomes. The daughter cell has got 8 chromosomes. So non-disjunction of all the chromosomes in mitosis in an early to an embryo doubles the chromosome number and produces the phenomenon of autotetraploidy. Now this is another pic. So you can follow either that diagram or this diagram. So here you can see it is having 4 chromosomes. Under normal conditions, what will happen is that the daughter cells, each of the 2 daughter cells will get the equal number of the chromosomes. That is, this daughter cell gets 4 chromosomes. This daughter cell will also get 4 chromosomes. That is when the normal mitosis occurs. But if this cell is subjected to colchicine treatment, so what happens is the colchicine suppresses the spindle formation apparatus, the spindle fiber apparatus. What happens is that in the daughter cell, these chromosomes, they cannot move towards the opposite pole, and in the same cell all these chromosomes, they come and lie together, and thus there is the doubling of the chromosome number. So this daughter cell has got double the chromosome number than the parental cell. Now let us take a typical example of what happens during meiosis. Okay, how autotriploidy occurs. We are going to look into the phenomenon of meiosis, where autotriploidy is going to originate, or how autotriploidy uh, cell is formed. So this is a diploid cell. Now it is having 4 chromosomes. Now, as I told that during the S phase, okay, during the S phase, the replication of DNA does take place and sister chromatids are formed. So each chromosome is showing 2 sister chromatids. Now what happens is as the cell moves on to anaphase first, non-disjunction. That is because the fiber apparatus is not formed in the daughter cells, all the chromosomes, they come and lie. Now this daughter cell has got double the chromosomes than the parental cell. Now when the cell moves on to meiosis 2, you can see now these gametes which are formed, these gametes which are formed, they are diploid. They are 2n. They are diploid. Now now, this is an abnormality. This is an abnormality because we started with the diploid cell and the gametes which are formed are also diploid. That is, it has resulted in the formation of unreduced gametes. Now, if these 2 gametes, they fuse with each other, then it will give rise to autotetraploid. Now, suppose if this diploid gamete, it fuses with a normal haploid gamete. If it fuses with a normal haploid gamete, then it will give rise to a triploid. That is triploid. It gives rise to a triploid cell. So, an autotriploid, that is 3N, it may arise when non-disjunction in meiosis produces a diploid gamete that then fuses with a normal haploid gamete to produce a triploid zygote. Alternatively, triploids can also be produced from a cross between an autotetraploid that produces the unreduced gametes and a diploid that produces the haploid gametes. I hope these points are clear. So this slide is representing the same explanation which I did before. Now let us see. Now in the previous section we saw how triploidy originates. Now let us next see when this cell undergoes meiosis, what happens. So in meiosis of a autotriploid, the homologous chromosomes, they can pair or they may not pair in 3 different ways. So this is a cell which is a triploid. Okay, it's a triploid cell. Now pairing can take place in 3 different methods. You can see over here what has happened. These 2 chromosomes have paired, whereas this has not. Now when this cell moves to anaphase first, these 2 chromosomes, they move towards one pole, other chromosome moves towards the other pole. Now when such a cell moves to meiosis second, so what has happened over here? you can see that there is a suppression of the spindle fiber apparatus, and you can see over here that in cell this cell itself, when it moves on to meiosis second, and then that is to anaphase second, you can see the gametes which are formed, they are diploid. That is, these resulting gametes are diploid, they are unreduced gametes. Whereas, you know when this chromosome, it moves on to one daughter cell. Now this takes place normally. When this moves on to the anaphase stage, then each daughter cell will receive one chromosome each. So these are the normal gametes. Now let us look into the second second possible phenomena. So what is the possibility is that all the three chromosomes may be involved in the pairing. So here also the segregation of the chromosomes of these three chromosomes, it takes place at random. So out of these three, this two may move to one daughter cell, one may move to one daughter cell, and this cell when it enters the meiosis 2, that is anaphase 2, it will form the normal haploid gametes, whereas this cell when it moves to anaphase 2, it will give rise to unreduced gametes. Now the third phenomena, okay, this is the third possibility where all the three chromosomes, okay, they're not showing pairing, there's no pairing at all. So here these chromosomes are free to move to any pole. So if by chance all the three chromosomes move to one particular cell, then what will happen is in the other cell there will be no chromosomes at all, and this will give rise to triploid gametes, and whereas the other gametes will not have any chromosomes at all, and this is lethal. So you have thus seen that in meiosis of an autotriploid, these homologous chromosomes can pair, or they may not pair in three different ways. So this is the same explanation. Now, now we discussed over here the mechanism of autopolyploidy. We saw how non-disjunction took place by by mitosis also. It also took place during meiosis. Now let us discuss this method, that is by genome duplication. So suppose if there's a cross between two plants which belong to the same species, two different plants but belonging to the same species, then what will happen is that the diploid which is produced, it will be fertile. Sometimes, occasionally, what happens is the chromosome number may double. That is, the genome duplication may take place, and that phenomena is called as the when the chromosome number gets doubled spontaneously without any external force, then this uh, the progeny, the offspring, it will become autopolyploid, and of course it will be fertile. So this is how autopolyploidy arises from genome duplication. What are the causes of genome duplication? It may be that there is meiotic non-reduction of gametes both in the egg and the male gamete also. Now, let us look into the applications of autopolyploids. So they are used in crop improvement. First, let us discuss about triploidy. So triploid seedless watermelons have been produced by hybridization between tetraploid and diploid lines. So here are some pics you can just go through. Even there are triploid bananas. Then you can see these are the pics not of tomatoes but they are of grapes. So these are diploid grapes and these are of tetraploid grapes. This is a pic of the oyster cells, which I will discuss a little in the next slide. So you have got um, triploid tomatoes, triploid grapes, bananas, apples, and pears. Similarly, we have got triploid sugar beets. Here, these beetroots, they have got more amount of sugar per unit area as compared to that of the diploid. So just that just in case you're very much curious about how the seedless watermelon arises, that's why I've added this pic. So these triploid, uh, usually these triploid plants, they do not produce the seeds. These fruits, they either are very few seeds or the seeds are completely absent, and they're called as seedless varieties. So we have triploid seedless watermelons, and similarly we do have triploid seedless grapes. So this is how the seedless watermelons have arisen. So this is for your extra knowledge. Now let us look into the applications of tetraploids. So tetraploids, they may be useful in breeding. They are useful in improving the quality. They are also useful in overcoming self-incompatibility. They are used in making distinct crosses, and sometimes they are even directly used as varieties. So some of the examples, important crop plants which are tetraploids are potatoes, peanuts. Peanuts means groundnuts, barley, maize, alfalfa, tradescantia, and even cucumbers. Um, there are seedless fruits like oranges, grapes, watermelons, and Macintosh apples. So apples are both autotriploids as well as autotetraploids. Similarly, grapes are both autotriploids as well as autotetraploids. So you can see over here watermelon, banana, grapes, then groundnuts and the strawberries, and this is the potato, which is sweet potato, which is an autohexaploid. Kindly excuse this wheat. Wheat is an allopolyploid. So tetraploid maize, it has been found that it has got more of carotenoid pigment. There's 45% more carotenoid pigment and more of vitamin A activity than the diploids. There are some even fodder crops, that is forage crops, for example, red clover, Trifolium pratense, then berseem, which has got high leafiness. The leaves are tender and they are softer. And it is found that there are commercial varieties of rye by name double steel and tetra pedicus, which are used, which are very popular forage crops. In banana also, autotetraploids are found to be disease resistant. So this is the pic of the phlox flower. In case of ornamental, it is found that the in tetraploids, the flowers are larger in size and they have got longer flowering duration, which is also called as long lasting blooms than the diploids. For example, in case of snapdragons, phlox and marigolds. Marigolds ಅಂದ್ರೆ ಚೆಂಡು ಹೂ ಅಂತ ಹೇಳ್ತೀವಲ್ವಾ ಅದು vegetas. Even in zinnias. Then autotetraploid cabbage and turnip are found to be larger in size and they have got more of water content. Even sweet potato is a hexaploid. Similarly, it is found that there are many medicinal plants which are tetraploids and they have got higher percentage of the desired phytochemicals. Example of animals are triploid oysters. Triploid oysters are edible and they are sterile. They do not spawn. So um, these are the favorite of those who like to eat the oysters. Then there are crabs, carps, which are weed-eating fish, and they are released into the ponds in order to control the weeds. Now, finally, let us see what is the role of autopolyploidy in evolution. So this pic is of the lilies. These are the lilies, which are also is an example of autotetraploidy. Now, AutoPolyploidy has contributed to a very limited extent in evolution of the plant species. Some crops like potato, peanut, coffee, lucerne, banana are found to be autopolyploids. Many forage grasses and ornamental plants are also autopolyploids, and autotetraploids they are found to be more successful as crops than others. Well, students, thanks for your attention.